couple of housekeeping, just a couple of housekeeping announcements for everyone. So first of all, you can use the chat and type in your questions at any time. We're going to watch the chat, and then also in between, we'll answer some of the questions. Also in the chat, you can upvote questions that are of mutual interest for everyone, and also please feel free to type in questions throughout the entire talks we're going to give. Links for further information on the call will be provided in the chat by our colleague Rita, who's organizing this webinar. So 38 participants, I think we can start. I will start with a quick presentation about what is Biomedx and how are we working. So let me quickly share my screen. Good. There we go. Okay, Rita, can you see my screen?
Yes.
Good, excellent. Okay, so let's start with a little bit of my background so that you know where I'm coming from and what my passion is. Technically, I'm a virologist. I did my PhD, finished my PhD in Heidelberg in the late 90s. And during my PhD, I started my first company called Multimetrics. I had a crazy value proposition at the time, 100 tests in 1 drop of blood. Failed miserably like many others later on, but I learned a lot. Then I joined a cell therapy company, which went up to phase 3 clinical trials with a liver cell transplant. Then I started a cancer institute called Heidelberg Institute for Stem Cell Technology and Experimental Medicine, which is still very successful on the campus here in Heidelberg, run by Andreas Strum. And then I was the head of the biotechnology region in Heidelberg for about 8 years, managing a public-private research and development fund at the interface between academia and industry. In 2013, I wanted to do something different, which is close to my heart. And this is when we started the BioMed X Institute. It's at the interface between academia and industry, and it's very much around my passion, which is helping young research talents to grow. And this is what BioMedx is all about. Technically, after we started in 2013, technically we are run like an independent biomedical research institute on the campus in Heidelberg. We're embedded in this campus, so we have access to all the different research core facilities, and we're combining the best of the two worlds, academia and industry. So on one hand, a lot of creativity, a lot of free-flowing ideas, sometimes crazy ideas, but on the other hand, also the know-how, how to translate that into something that will help a patient sometime. And we do this using a unique innovation model, which is combined of global crowdsourcing and local incubation. What that means, I'm going to explain in a couple of minutes, but we've published this new innovation model in 2015 in Nature Biotech. The way it works is that most of my time I spend with people like Thomas. I help big researchers in big pharmaceutical companies that want to think far into the future, to ask very big questions. Big questions that always start with what if? What if we had a platform that could help us to develop, for example, a drug in a much more efficient and in a faster way? That it will help patients much sooner and it doesn't cost that much money. And once we have identified such questions, we call them challenges, we post them on our crowdsourcing platform worldwide at the best universities and research institutions, and invite scientists to apply by submitting a very original project proposal, how to solve this particular problem. These are very short proposals, 2, 3, 4, 5 pages. What's important is that it's a very original approach how to solve the challenge. And then usually we get between 100 to 200 proposals, sometimes up to 500 or 600 from 60, 70, 80 countries. So it's truly global. And then we invite the 15 most brilliant scientists to a 5-day bootcamp to Heidelberg, where they see each other for the first time. And then we help these scientists to associate in 5 different groups, and we help them to convert this collection of sometimes crazy ideas into something we would call a truly brilliant project proposal. So all these talents in this 5-day bootcamp learn in a very quick way how to prepare the perfect pitch presentation of a really, really ambitious research project. And then on the last day of the bootcamp, they present in front of the jury, which is usually the senior management of the pharmaceutical company we're working with. And then we jointly select the winner, and then the winner is then moved with his or her family to Heidelberg for a project term of up to 5 years in our institute, with access to all the core facilities of all the main institutions on campus. Also, we have a fully equipped lab. And of course, these teams receive very intense mentorship by our mentors from industry, but also from academia, making sure that we are producing something truly outstanding. But we also publish. That's also very important for us. This is the atmosphere you will see at one of those boot camps. This is one that was in more or less a year ago with Merck in the field of extrachromosomal DNA in cancer. What's very important, especially for me as the founder of the BioMed X Institute, is our core values. And this is also why we're doing these boot camps. First of all, our first core value is cross-leverage. We inspire and motivate each other to unleash our full potential. What does that mean? We want people, scientists, who have embraced the concept of growing by helping others. So as you will find out, Biomedx is an environment where all the scientists help each other, where they share their ideas freely, and where they support each other. And this is very important for us, for our ecosystem. Second is cross-pollinate. We embrace diversity as it is the source of innovation. In other words, the more people from the more different perspectives look upon the same problem, the higher the probability that innovation will happen. And if you visit us, you will see that people are very diverse. We are coming from all different backgrounds. We're all very passionate about science, but we're very different, and it's good that way. So you will see that diversity is very important for our ecosystem. And third, aim high. Our goal is to enable a new generation of targeted therapies. So we expect also, we provide a lot of support for all our research fellows, but we also expect that you make the most out of it for your personal career, so that after the term at the BioMed X Institute, you can start into your next dream position, either in industry, in a startup, or back in academia. We've done this in the past 9 years in Heidelberg with 6 different pharmaceutical companies: AbbVie, Boehringer Ingelheim, Johnson & Johnson, Merck, Roche, and Sanofi. And in the last 9 years, we have started 21 research groups in Heidelberg. 10 projects were successfully completed. And what's also part of our model is that at the end of the project, our pharma partner has a first right to acquire the results and then continue the project inside. And our success rate so far, 8 out of the 10 projects that were finished were actually then taken over by the respective pharma partner. We have developed quite a significant biology expertise in oncology, immunology, and neurosciences. And in the next slide, I'm going to give you a little more overview of what all our teams are doing. So most of our teams are working with human materials, primary human materials. They're trying to replicate what's happening in the human body in very complex organotypic culture models. So from intestinal epithelium, synovium, lung epithelium, different brain, different kinds of tissues. Second is this is combined at Biomedx with multi-omics and artificial intelligence and machine learning. All those models generate huge amount of deep biological data, and it's very important that we analyze those data well and that we draw conclusions and generate insights from these data in a very smart way. And also there's some teams that are developing actual sensors, which then, of course, are also helpful for all the other teams. So you have to imagine all these teams in these different areas, they help each other and support each other. Of course, this is not in the middle of nowhere. This is in Heidelberg. On the left side, you see about a map with about 1.5 hours drive radius around Heidelberg. We're right in the middle of it. And as you can see, there's lots of biomedical innovation going on, lots of big pharma companies with R&D sites, including Sanofi in Frankfurt, and 9 universities and research institutions. And the centerpiece, as some of you might know, is our campus im Neuenheimer Feld in Heidelberg. This is a little more than it's about a square mile with more than 100 different institutes in biomedical research and medicine, representing 22,000 jobs in research plus 16,000 students in the same field. So it's a very high density, very dense ecosystem, and our fellows, of course, are closely interacting with most of those institutes. We are based in Technology Park, which has a total of 40,000 square meters of lab and office space for companies, and we currently operate 1,800 square meters fully equipped lab space in the Technology Park. Also, our fellows, through framework agreements, have access to different large research institutes like the European Molecular Biology Laboratory, the university and the hospital, and also the Cancer Research Center. And this includes animal facilities, large instruments, and of course, a lot of academic mentors and PIs with whom we collaborate. At the moment, our teams had over 60 collaborations with major institutions worldwide. We are really focused on deep research, deep science. We have published more than 50 papers in peer-reviewed journals in the last 9 years. This is also important for us because some of our fellows, after the time at BioMedx, they go back to academia and become professors. Now, since this is working in Heidelberg, this is just a quick excursion. This is working in Heidelberg. We also thought, can it work somewhere else? And since last year, we are also present in Israel, right next to the Weizmann Institute in Rehovot. We have started together with 4 different pharma companies, Merck, Pfizer, Teva, AstraZeneca, then also Amazon and Israel Biotech Fund. We have started Ion Labs, which is a slightly modified model. There we create startups and not research groups like in Heidelberg. Also, we focus entirely on artificial intelligence for drug discovery and development. And we have a situation where multiple pharma companies and partners sponsor the same project. And we have now enough money to start 20 startups this way using our model. In Israel this year, we have already started 4. I just came back from Israel together with Rita, who organized the bootcamp in Rehovot. Last week was very intense, but also we had a great winner, and we're going to start a new company there soon. And in addition to that, this is just an announcement. Early next year, we're going to start on the campus of Yale University in the US, in New Haven, with 2 projects. So we're also expanding to the US. Now, coming back to the activities, if you join BioMedx in Heidelberg, what you can expect. We put a lot of effort into the ecosystem. There's a lot of engagement programs like special interest groups in lab tools and methods and career development. We focus a lot on science. We have an instrument that's called the Friday meetings, where all our scientists are in a room. At the moment, it's roughly 80 people. They're in the same room. And the group leaders decide who presents his or her work in front of everyone, and then everyone helps and troubleshoots and contributes. And also we have an annual science retreat where my main job is being the DJ and also the bartender at the karaoke session. We put a lot of effort also in education of our scientists. We have a scientist entrepreneur course where you can learn how to start your own company. Scientific writing courses, German classes. Heidelberg is a very international place. You don't have to speak German. Many of our scientists never learn German, but if you want to, you can, of course. We're interacting with the campus, lunch talks. Once a month, we invite the top speaker from outside and then invite the entire campus in Heidelberg. And also we do sports together, charity runs, dragon boat races, climbing, yoga, and also a lot of social activities. So potluck, we bring food from our home countries. At the moment, it's around 25 different countries at BioMedx. We have a beer hour, which is organized once a month by one of our groups. Annual summer party, beach party, which is really a highlight every year, and the year-end cooking event where we jointly cook food and then eat together. And so far, we didn't have a single diarrhea event, so that went well as well. This is just some photos of the fun part of the activities. On the left side, you see the last dragon boat race where our team was Team Smurf, but also the scientific retreat, which was a great highlight. And also, of course, we celebrate the PhD defenses and also master and bachelor defenses of our students at BioMedx. And of course, we enjoy karaoke and German cuisine. Value proposition for talents to come to BioMedx. First of all, of course, it's a 3 to 5 year fully funded research group. In the case of this call with Sanofi, it's an annual budget of 950,000 euros for up to 5 years, so up to nearly 5 million. You can focus on outstanding research and publications, so no teaching obligations like in other academic positions. It's a very creative cross-cultural teamwork atmosphere. You get guidance from experienced mentors from academia and industry. It's an outstanding environment with direct access to the leading research institutions in Heidelberg, and also you can have leadership and entrepreneurship training. And all in all, it's very important. It's a risk-free assessment of a potential future career in a startup or big company. If you like it, about 50% of our fellows transition to industry, and the other 50% go back to academia because they have a continuous publication record. So each of our teams usually has between 4 and 6 publications in peer-reviewed journals. The group leader is always last author, and almost all postdocs and PhD students finish their term with at least one first author publication. Where do people come from? Can we attract top talent from around the world? I think after 9 years and nearly 300 alumni, I can say yes, we can. This is just some of the logos where people came from before they joined BioMedx. And where do they go afterwards? I mentioned already industry. Of course, a very obvious potential future career is with the sponsor of the project, but also sometimes teams then are hired by a different pharma company. And as you can imagine, you will start your career with a very strong network to senior management of those companies, because you will see them in several times a year. And yes, this is my part. So I would like to finish with welcoming you to our Biomedx family. With this barcode or with this link, you can register on our career space. If you register, even if you don't like the topic of the current call, if you opt in, you will get an email whenever we publish a new call for application, either in Germany, in Israel, or in the United States. So you can stay up to date, and you can reapply anytime just using the same profile. And of course, we would like to encourage you to follow us on social media, on LinkedIn, on Twitter. Personally, I like to connect with early career researchers. If you send me a request, an invite, I will always accept if you were part of the webinar. And also, of course, you're welcome to follow our institute. And with that, I close my presentation. And let's maybe quickly check for immediate questions before we move to Thomas. Rita, do you see anything urgent?
No questions yet.
Okay, that's fine. Then I hand over to you, Thomas.
Yes, thanks, Christian. Let me share what I have here. Can you hear me well?
Perfect.
Good. Okay, so thanks again, everybody, for joining this webinar and for interest in learning more about this Sanofi BioMedX challenge. I'm championing this project at Sanofi, and in the nomenclature used by Christian and BioMedX, I'm the industry mentor for this program. And actually, I'm today very happy to provide a few more insights and together with Christian, answer a few of your questions. Okay, so this is the plan for the next 20 minutes. I would like to introduce you to Sanofi, how we are chasing the miracles of science to improve people's lives. Then I'll introduce myself, who I am, what I do, and what's my motivation and purpose. And last but not least, I will zoom into the problem we are facing, share with you a vision of where we would like to be, and present you finally the challenge, and the challenge that is really then meant to find a solution that is taking us from where we are today to where we want to be. Okay, so let's start with a few slides on Sanofi. Probably some of you have already an idea of what Sanofi is about and what we do. And the next slides will get everybody on the same page. So Sanofi is present in about 100 countries. We do have about 100,000 employees. I'm one of them. We are producing our medicines and vaccines at about 70 manufacturing sites. And more important, obviously, for this call, we do have about 20 R&D sites. So this is our motivation statement for Sanofi. We are striving to develop breakthrough medicines and vaccines. All we do is fueled by data, digital technologies, and cutting-edge science to really transform the practice of medicine, and actually also to turn the impossible into the possible for millions of people around the world. And a few words on our ambitions in R&D. So we focus on a few priority therapeutic areas. And in order to invent novel medicines for these key therapeutic areas, we do have a battery of discovery platforms and modalities. And I will zoom into these in the next slides. Our investment in R&D last year has been quite significant. As you can see here, Currently, we do have 91 drug candidates in clinical development, with 70% being biologics and 80% being either first in class, meaning they address a novel mechanism of action, or really aiming to become best in class medications. So what are the therapeutic areas of interest for Sanofi? So besides vaccines, the main flagship TAs at Sanofi are immunology, inflammation, and oncology. Other focus areas at Sanofi are neurology, rare diseases, and rare blood disorders. And in case you are interested to learn more about our actual R&D pipeline, please visit our Sanofi webpage. As mentioned before, Sanofi is using a battery of different platform technologies to drive our R&D pipeline. And these platform, or you can also call them modalities, include more traditional approaches like small molecules and enzyme replacement therapies, but also include a variety of novel antibody approaches, cell therapies, gene therapies, siRNA approaches, or messenger RNA approaches for vaccines, but also for novel therapeutics. So in the context of today's call, it's also important to mention that Sanofi is developing an emerging leadership in immunology. So we do have several products on the market and several clinical development candidates in our R&D pipeline for the treatment, for example, of inflammatory conditions like rheumatoid arthritis, psoriasis, and asthma. And this is also why we have chosen the immune inflammation space as the focus area for this Biomedx Sanofi challenge. So as said, for this Biomedx Sanofi challenge, I'm honored being the industry mentor for this project. And I would like to spend a few minutes also introducing myself, who I am, what I do, and what's my motivation. So this is a snapshot of my personal career journey. I'm now with Sanofi for 23 years and have been in various roles and functions. I started actually in 1999 in the area of molecular modeling, where I was designing small molecules by doing computer-aided drug design. My PhD actually is in biochemistry and structural biology. And then in between 1996, 1999, I've been a researcher in the US, first a postdoc, then a research assistant professor at Texas A&M in Houston. Actually, during my time there, we had a project to design small molecule inhibitors for some amyloid diseases in humans. And actually, looking back, I believe that this project really woke my interest to move to a pharmaceutical company like Sanofi, really to discover new medicines for patients. And so after 3 years in the US, I returned to Germany. I took a position at Sanofi in 1999, starting in this molecular modeling team here in Frankfurt. And then actually in 2010, I was given the opportunity to build a small team. This team was called Computational Biology and Bioinformatics. In these days, we started to look into the dynamics of the processes linked to the secretion of insulin in the pancreatic beta cell, because we were doing insulin research. And then actually in 2016, my team was moved from research discovery into clinical development. And actually this allowed me and my team to gain experience in the clinical development space of pharma. In 2018, I became what we called head of translational informatics. 2019, the whole group was moved into a new organization Sanofi was building called Data and Data Sciences. And in DDS, I'm now actually heading a global team called Translational Disease Modeling. So I would say in summary, during my career, I've been into several areas of data sciences, starting from structural biology to molecular modeling and in silico drug design, from bioinformatics to computational biology and now quantitative systems pharmacology. I've seen the world of discovery research, but also the world of clinical development. And along my career journey, I actually have moved from a contributor role, became a manager, and now I'm actually a leader and a coach. So what's really inspiring me? So this is the vision that actually one day we can test drive all our development candidates in virtual patients. So really to explore if there's a strong reason to believe that these development candidates will show best in disease performance in real patients. And actually to really decide if it's worth taking them into the clinic, because this is a very critical decision in pharma. And also these virtual patients should provide the knowledge and insights needed to allow us to choose the right dose and to select the right patients for the clinical trial. and also to identify those patients that would benefit most from this novel treatment approach. So that's what I'm looking at. That's why I'm getting out of bed every day. My role currently, as I mentioned, I'm global head of translational disease modeling, TDM. We are organized along the 3 main therapeutic areas. We are a team of 20 scientists across 4 different sites. Actually, most of my people have a PhD in biomedical engineering. We also have a few pharmacometricians and mathematicians. So I can claim we are truly translational function supporting actually projects along the whole value chain in R&D. And actually what we do is we provide computational models of disease biology, disease progression, and also the known pharmacology. And these models then can be applied to gain insights into the patients we want to treat, also our drug candidates, and they can also be used to perform simulations, meaning that they can be used to explore the expected clinical efficacy for some of our drug candidates. Okay, so let me now introduce the problem we are facing on our journey to discover novel medicines. So what is our vision of where we want to be, and what's the challenge we would like to pitch to you? Okay, so let's first define the problem in the discovery process for novel medicines. So I don't know who from the call is already somehow has been exposed to what pharma is doing, so I start here more or less from scratch. But in this cartoon, actually the drug development pathway is shown in 4 stages. Let's just focus on the first 2. So the first stage is labeled here in vitro and in vivo testing. So in this stage, novel drug targets are discovered. Actually, novel drug candidates are made and tested for their biological activity, often in human in vitro models or in preclinical animal models. And then when the right development candidate is identified with the potential to become a new first-in-class or best-in-disease medication, the asset is then moved into the clinic. And then the second phase starts, the clinical development. And here, actually, the drug candidate is tested in humans in clinical trials. First, classically, you start with a phase 1 trial to explore safety and the pharmacokinetics, the PK of the molecule. Then there's the second, the phase 2 trial to explore the clinical efficacy in a smaller patient cohort before then finally the phase 3 trials start to explore the clinical efficacy in a larger patient population. So now comes the problem. So at the stage of the transition from preclinical to the clinical development, really a model or tool is needed that would allow to reliably predict the clinical efficacy for a novel drug candidate. So at this point, we need to know, is there a quantitative reason to believe that this asset will really become best in disease? Will it show better clinical efficacy than competitor molecules that are already available on the market? Is there maybe also a specific patient subgroup that would benefit most from this treatment? And last but not least, we also would like to know what could be the markers, the biomarkers that would allow to identify those patients that would be expected to show optimal treatment response. So obviously, I mean, pharma is existing, so there must be already solutions today. So, and I would just like to zoom into 4 approaches that are used to support this, what we call clinical translation. So all 4 are currently applied, but each of them actually comes with an advantage, but also with a disadvantage. So first we have in vitro models, for example, cell lines expressing the target receptor. These then allow to actually test the biological activity of a novel drug candidate. They can also be tested then what we call in ex vivo models, for example, blood or tissue samples taken from the patients. Then second, there are preclinical animal models. These can provide information on the pharmacokinetics, the PK and the PD, pharmacodynamics of a new drug candidate. And what is then seen in animals can be used to predict the human PK and also the clinical PD effect in humans, especially when the chosen animal model is similar to the human disease situation. Third, also systems biology approaches can play a role. Here, genome-wide data sets of patients are used actually to identify gene expression signatures, to identify patient subgroups that would be expected to benefit most from a given treatment. And actually, although these systems biology approaches have a good track record to support patient stratification, they are currently not systematically applied to support the prediction of clinical efficacy for novel molecules. And last but not least, we have quantitative systems pharmacology modeling, short QSP, which is using all prior knowledge of human physiology and pathophysiology to generate virtual patients. And actually these computational models, or we call them virtual patients, allow actually then to predict the expected clinical response in humans. However, and that now comes the limitation. The limitation of this QSP approach is that QSP is only considering those biological pathways and processes that are already known, that could be quantified with respect to their dynamics, and that have been included into these QSP models. So the flaw here, the limitation, the disadvantage is that QSP is usually not covering the unknown. Okay, just 2 slides on what QSP is. So this is actually a short definition taken from a review article published 10 years ago. So let me just quote, it's a quantitative analysis of dynamic interactions between drugs and biological systems. Important to mention is that a QSP model is based on so-called ordinary differential equations, ODEs, that describe the dynamics of relevant biological processes. It's in this way a dynamic modeling approach capturing longitudinal and time series data. Actually, QSP sits between classical PK/PD modeling and systems biology, as shown here in the table. And maybe you know PK/PD is traditionally done to empirically evaluate the PK/PD relationship for a drug. And on the other hand, we have these systems biology approaches that use actually clinical genomic data sets to unravel unknown connections in biological pathways to support target identification, sometimes patient stratification. So let's zoom a little bit deeper on that slide into what kind of data is used to build a QSP model. So a QSP model integrates a lot of different data sets and knowledge into a single computational framework. First, the QSP model integrates a lot of prior knowledge, mechanistic descriptions of physiology, disease biology, and pharmacology. Second, it integrates then the available clinical trial data and legacy clinical studies for the indication of interest, as well as also data from clinical trials from competitor drugs. Third, it also integrates then the human in vitro data from the drug candidates and the competitors of interest, like data from binding or functional assays, also, as mentioned before, from whole blood assays. And then fourth, it also integrates the PK data for the drug candidates and the competitor drugs of interest. So the computational model thus can be seen as a virtual patient, and that can then be used to gain insights into our patients and drug candidates, and also then to explore the expected clinical efficacy of a drug candidate in virtual patients. However, as mentioned before, there is one limitation or disadvantage of the QSP modeling approach. This is that the model is only capturing the biological processes that are already well known and that actually can be quantified in these ordinary differential equations. So in a way, QSP is not seeing the information that may sit somewhere in undigested, large genomic-wide or clinical genomic data sets. And actually, obviously this information is important because it can hint to unknown connections in biological pathways. So thus, although QSP modeling can be successfully applied to assess the clinical efficacy of drug candidates that act on known pathways and biological targets, it has some limitations in predicting the efficacy for drug candidates with a novel mechanism of action. And these are what we call then first-in-class assets. That's what Sanofi is mainly interested in. Okay, good. So here's now our vision. So imagine we would have virtual patients that would allow us to test drive all clinical development candidates we have, including those drug assets that do follow a novel mechanism of action in virtual patients, and that would then allow to predict their efficacy in clinical trials, even allow to identify the right dose and the right patients, and all this based on, well, the virtual patient information, the knowledge on these patients, and some information we would need to feed in from the drug candidate from in vitro and ex vivo profiling. So here's a little mock-up of how this process could look like in action. So let's assume we do have a development candidate. Let's assume it's a bispecific nanobody for rheumatoid arthritis, and it's meant to modulate 2 key cytokines in inflammation. Let's call them A and B. And let's assume we know the expected human exposure of this nanobody and its binding affinity to these two human cytokines A and B. So we would feed this information into this virtual rheumatoid arthritis patient and then actually simulate the clinical effect of this drug candidate for a given dose. And what we then want to know is how would the application of the drug candidate shift the RA, rheumatoid arthritis disease score, in this population compared to a competitor molecule that is already on the market. So we would not only like to see the difference in the average response for both treatments, but really would be interested to see a distribution across the virtual patient population. And in addition, we would like to learn which are the patients responding best to this novel treatment and what may be the markers to identify these best responders. Okay, now you have an understanding of the problem, I hope, and also the vision where we would like to be. And here comes now the challenge. So we need your skills, your energy to think about an approach, how to build the next generation virtual patient engine that would allow for making this vision to become a reality. So we don't know which methodology you would like to consider for tackling this challenge, but probably you would like to, you want to think about a combination of a QSP type and systems biology approach, and in addition, blending in data from in vitro or ex vivo profiling to ensure a really good prediction accuracy. So we are looking for a virtual patient engine, as mentioned before, that is able to predict the clinical efficacy for novel first-in-class therapeutic approaches. I'm getting to my last slide. Actually, a bit more on this challenge, just a little bit more detail. So what we are looking for is a disease-agnostic approach or engine. So an approach that is working for any kind of disease area. So obviously, the engine will need to be built and run with data, and Sanofi will provide access to preclinical in vitro and ex vivo data. Also, we will provide access to clinical trial data for reference drugs, and where available, also to molecular level patient data and real world evidence data. And as mentioned, although this engine should be disease agnostic, so should work for any kind of disease area, in this project, we are really striving for a proof of concept to generate an engine for a defined indication from the immune inflammation space. Could be atopic dermatitis, could be inflammatory bowel disease. This needs to be decided still. It also depends a little bit on your experience and expertise. So, but it should be, the engine should be test-driven or built as a proof of concept for an indication in the immune inflammation space. You will have to identify the optimal approach, what type of data would be mandatory for successful prediction of clinical efficacy, possibly identify the need for additional wet lab data that would improve the prediction accuracy that would need to be measured in a wet lab experiment. And you also would have to validate the approach and finally use the engine to predict the clinical efficacy for some Sanofi drug candidates for this indication. Actually, this is the last slide now. So a little bit of a zoom in, and maybe Christian would like to comment on this a little more. So what kind of profiles are we looking for? You would need a master or PhD degree with an outstanding track record and a strong interest in this field. You would need to be thrilled to lead or collaborate in a cross-functional team of wet lab, data, and computer scientists. For the group leader positions, you need certain experience, 4 to 8 years postdoc. And for the postdoc positions, you need a PhD or master degree in data science or molecular biology, cell biology, or pharmacology. So we hope that you are now as excited as we are about this project, and we are happy to take your questions.